talk a little bit about what are some of the challenges and opportunities, especially applying this type of skill, the technical computational programming, data science skills to big data. So the first thing that I would start with is that surprisingly, we have access to a lot of high quality data sets. Not only are these data sets generated by experts in excellent labs, the data sets continuously update. So we have new and more detailed understanding of different areas of life sciences through the lens of these data. For example, just in this pandemic, we have a tremendous growth, not only in the quantity and quality of data, but also the rate of data submission. So if you compare this to previous pandemics, like the SARS-1, Ebola virus, MERS, or Zika, this pandemic has generated so much data in almost real time and has made that data accessible to almost anyone that would like to. Now the question is not so much about data availability, but what to do with this data to extract meaningful insights. And that's where I think these skills really come together. So first of all, data could be of many different types. It could be imaging data, clinical data, streaming data, omics data, chemical, phenotypic. There are really many different types of data. But for all of them, one of the first challenges is to create structured data that you can actually work with. And so to do that, the first thing that we will talk about is how to take these different types of omics data, including phenomics, phenotypic data, genomics, variant data, epigenomics, different types of histone modifications, DNA methylation, transcriptomic data, gene expression, and things like that, and how to take that information and start processing it, creating structured tables. And those tables could come in different formats. They could be matrices with just numbers. They could be data frames with both numbers and different types of levels for factors or names. And now that we have this structured data, now the second challenge is really to be able to ask the right kinds of questions about this data, right? Nobody just takes data and structures it so that you can, visually see what's in it, right? Everybody works with data to be able to answer some important questions. And that's another part of data science that I think differentiates a data technician from a data scientist. The science is really all about asking questions that are supported by evidence. And so the data exists to help us extract evidence from the data set, but for us to have a relevant question, what we want to do in this program is to introduce you to some of those critical and important and interesting questions that could be asked of these types of data sets so that you can really utilize the data effectively. And so what we hope that this program can provide for you is a complete roadmap for data science that will be an application to biomedical research data, but it will include things like finding different data sets, learning how to explore and visualize these data sets to understand major characteristics of the data, how to understand different types of research methods that others use and extract the evidence that you need to answer a research question, and ultimately how to showcase or illustrate what you have found with relevant methods of insights so that you can visualize your data, so that you can identify the properties, the statistical properties to point to, and ultimately lead to understanding. So we call this program Omicslogic because really it combines the omics data and the practical skills with a logical approach. And it guides you from A to B, from knowing about all of these terms and wanting to apply them to actually understanding of how to apply them and then demonstrating what you have learned. So we'll take several different types of projects using biomedical omics data, from diagnostics in cancer biology, single cell and tissue composition, microbiome and infection. And in the context of these different types of topics, we will learn about your typical data analysis skills that you would have to learn to use effectively to extract meaningful insights, including data collection. So how was the data collected? What does it represent? Data cleaning. How do you extract only the relevant information or the good quality data, right? and make it tidy and ready for analysis, how to perform exploratory data analysis, and how to iterate that process so that the insights come from the data, data-driven questions and hypotheses that you can generate, and then how to explore the data in a way that you can really apply statistical modeling and inference from those models so that you can get meaningful results. And obviously, a big part of that is going to be focused on data mining and classification. So we will learn about different types of unsupervised and supervised techniques that have been used extensively on their own, but also as a part of meaningful bioinformatics work. And the principles of data science that include statistics and data wrangling are going to be very meaningful here when we go to machine learning, because you will essentially operate with an extraction from different portions of the data to train your model, to validate your findings, to make sure that your model is accurate. And you will learn to differentiate between different types of methods so that you can compare and contrast the results and select the ones that are most appropriate to the type of problem that you're solving. Now, the same methods are also included in many different types of biological data mining, and those include things like differential gene expression or principal component analysis and standard tools that are adapted from these methods to address more and more technical variation and complexity in the types of data that is being collected, including integration of multi-omics data, but also including more and more advanced data types like single cell data and data that combines multiple elements together. And so that's why we really are going to be looking at a variety of applications for multi-omics data integration and integration of this omics data with clinical data using the different examples that we will try. So to give you an overview of what this program is going to look like, I would like to take you to the program page. So for all of you involved, sorry, let me go over here. Okay, so everyone that is interested to join this program, the easiest way to show you what this program is going to include is to take you to the program page. So this is a program page. It is called LBRN Omicslogic Data Science. And so I'll just explain what does LBRN mean. LBRN stands for Louisiana Biomedical Research Network, our major partner in this program. And because of their support, we are able to make this program more accessible to more people. And so the types of information that you will get on this page are going to, I think, address many of the questions that you might have joining today's webinar. So first of all, the program is going to start on July 12th and last for a month. And during this time, we will cover a number of different topics by participating in live sessions like today, where you can interact with the mentors and get answers to your question, as well as practice in class to perform different types of analysis. You will also be able to use asynchronous resources in between our sessions to gain more technical skills at your own flexible schedule. So in many cases, we will go through a portion of the material by going through them in our session, and then we will point you to resources that you can supplement this knowledge with by performing actual technical steps of writing the code, trying to run the code, getting the outputs, and even troubleshooting some of your results. So to illustrate how that part of it will work, let me ask you, how many of you already have an account on Omicslogic? So please, if you do have an account, maybe you can put in number 1 in the chat. If you, okay, great, thank you, Leanna. If you don't have an account, just take a minute to register. The way you register is you go to log in and you create an account right here on this form. After you've created the account, you will have access to the coursework that we will discuss. Okay, great, excellent. So I see that a lot of you already have this. And once you're logged in, you will be able to see, yes, you will be able to see some of the courses that are going to be included in this program. Okay, so is there anyone here that have not registered yet? Is there anyone here that has not registered yet? Okay, if you could take a moment and register, please. We'll give it just a couple of minutes so everyone is able to follow through some of the examples that we will be going through. Okay, so once you are logged in, you will be redirected to a page where you see a collection of courses that are linked to your profile. And what I will demonstrate is how we will leverage some of these resources to actually practice what we are going to do in this program. Okay, so I see that someone is raising their hand. Would you like to ask something? Okay. Okay, so if you're already inside on the course page, please search for data science, and you will see here a couple of courses: data science, introduction to data science in Python, and introduction to data science in R. Okay, so once you are in these courses, you will be able to see what types of methods we're going to be covering and even try some of them with a free registration. So if you register today or you already have been registered before, you can access all of the free courses. And then when you complete your registration, and there's a number of ways that you can register that I'll talk about, you will see courses with this star, and those would be premium courses that address some of the more advanced topics and require some more practice. So you'll be able to access those with a premium subscription. One of the things that is always difficult with large, complex data sets is to grasp the type of information or the type of relevant information that is in the data quickly, not spending a lot of time on individual feature like a gene, and more looking for general trends. So what I want to show you today briefly is an example from a course on dimensionality reduction, principal component analysis, and visualization. Now, principal component analysis is most useful for continuous data, which means that the data is in a numeric form, just like you see inside the cells over here. And it could be anywhere in a continuous scale from 0 to whatever the maximum is. And it doesn't even have to be 0. It could be also centered. So it could be -1 to 1, or it could be negative and positive numbers. But the key point here is that it is continuous data. And so the continuous data is represented here in this example as a gene expression profile for a number of different groups. As you can see right here, we have a row for groups, and we have sample IDs, which are essentially names of cell lines that have different properties. So they come from different breast cancer patients. And so using this example, what you'll see in this course is how you can apply a principal component analysis technique to quickly evaluate the separation of samples into different groups based on this gene expression profile that is found inside the data matrix, which is just the numeric portion of this table, of this data frame that we were looking at just a moment ago. So you have a brief video that you can go through to really understand the challenge of the process to getting to this outcome, but also you have a breakdown of the full code. Now, for many people, including myself, one of the main challenges in learning data science is that most of the courses out there start with very basic things like learning how to print out hello world. In our case, we're assuming that you are ready to jump beyond that and start with a data set that you understand. And that's why we've selected these data sets that come from a background relevant to life sciences. And so I think many of the people are more motivated to continue and learn about a method once it's talking about a data set that you understand. So in this case, this is gene expression for cell lines, and those cell lines could be categorized in groups based on their type or subtype of breast cancer. And so the first thing that you will see here are some important parts of the code that you have to master for any kind of a script. And in this case, we have different libraries that will be used. And these libraries include libraries which are packages that include many different functions that we can pull from to actually apply to the data set that we load. The second line here is going to be how to load this data. So this data exists here on the internet, but how do I put it into the script, right? And so that will be there. And then we start working with this data set to extract only the meaningful portion of the data for our analysis, and then launch the analysis. Now, once you learn these brief steps, it's very easy to understand how we can apply the same technique to many different types of data and how to start working with the tables. So here you see two different methods, the base method in R and the ggplot method in R for visualizing this data. And as you go through, some additional useful information is going to be added to the plot, like labeling the plot, or even projecting the feature significance or how individual genes contribute to this principal component analysis plot, a two-dimensional plot that we're looking at, so that we can evaluate which genes are actually responsible for the separation into different groups. So once you go through some of this basic explanation in learning the code, you will be able to try the code yourself. And trying the code starts from just simply pressing this button, run. Okay, so just by running this code right here in the browser, you already get some output. And you can play with this to change things so that you can see what is actually going on. For example, as you're going through this code, you can see that we can there are actually two different methods to visualize the PCA output. So here you have the basic method with plot, and here you have a ggfortify, so a library to do everything in a different approach. And simply by deleting this, we can try to run it again and only get the basic plot, right? So now we can separate the plot into portions that we understand. So we understand what the first portion did, we understand what the second portion did. In order for me not to forget, I can just reload the full script and get all of the things that were in there before. So I can actually now remove this part of it right here, run it again, and only get the ggplot output, right? So now I'm working with two different libraries. One is the base library, including in R, and another one is a library called ggfortify that I can actually look up online and see what does it do, right? So I can start looking into all of the different options for ggfortify and see what does it do. And many of the experiences that you will have applying data science is going to be related to looking up different packages and seeing what kinds of additional methods are going to be used and how are these methods actually used by others to use, for example, for principal component analysis. So you can always find these packages on their different repositories online. Okay, a question from Abu Bakr. Can you run the code with your own data? Excellent question. So this is a code that is going to work anywhere, right? So simply taking this code right here and copying it to your clipboard, you can now install R and RStudio on your own computer and run this code on the data set that you would like to analyze. However, let's take a look at how this data was loaded. You can see that the input file is given via a URL. This is a URL to a GitHub repository where we store all of the data sets that we'll be going through. On your computer, that data set is going to be sitting somewhere else. It's going to be in your folder, but in a very similar way, you're going to be using the path instead of a URL to a public domain. You'll be using a path on your own computer. And you can do that by setting up a working directory on your computer and then linking to that directory, not through the full path, but just giving the name of the file. So that's something that you can try. And you can also try some of these downstream codes that are going to be much more detailed in what kinds of outputs they provide you with, ultimately leading to a interpretable result. Okay, so let's take a look here at the final result of this analysis. You can hover over the individual dots that represent samples, and you can see that they're colored in four different colors. There's the luminal, which is pink, basal, which is green, normal-like, which is blue, and cloud and low, which are dark blue. Now, what is this box that we can spin around? This is essentially a space defined by three principal components: the X, Y, and Z. Those are axes in this space, or principal components selected by the top information that they carry based on how much percentage of variance they show for our data. And so just by learning about these types of outputs, we can start asking questions about similarity and differences between these groups of samples, and how we can actually take a classification model to train an algorithm to identify the samples that we see here based on feature significance, based on specific genes that are in this data set. And so going step by step through examples that you can run right here on the screen to more and more and advanced methods. You can see here, going beyond the basics, we'll introduce some additional applications of statistics, for example, to understand confidence levels on this principal component, and eventually turning this into a data science problem. And the data science problem is going to basically tell us that I have a lot of data, I have mined this data, and I have identified some differences between groups that I can interpret clinically using phenotypic information. And now I want to address a problem of finding these types of subtypes in a different data set. And so now we can really translate this into an algorithm that will look for specific markers that will identify those types of patients or cell lines that match a certain clinical phenotype. So as a brief introduction to data science, I think this is a good example that shows us how we can apply this data-driven approach of data mining, classification, training a machine learning model, and asking the right kinds of questions to understand biology and to be able to leverage these signal points to really address a major problem, which is classification of different types of cancers, especially based on biomarkers. So genes that we can detect and we can then use on big data to mine those patterns. Okay, so if there are any other questions, you can go ahead and put those questions in the chat box. And in the meantime, I would like to return briefly to our presentation to talk about how the other components are actually going to be used. So as I mentioned, this is going to be a program that combines live sessions like we have today, where you can ask questions. We can go through code together and try to run it, maybe on our browser code playground or maybe on your own computer, compare the outputs and be able to ask specifically whether we understand them with self-guided study materials. So materials that you can then practice online independently and be able to validate that you can actually go through these questions, go through the same types of challenges and examples, and then receive practical assignments. So in these sessions, we'll give you some assignments to complete. So you can try it, practice it, and then test whether you really understand them. Okay, so at this moment, I would like to take a pause and launch a poll so that we can get a better sense of who's in this session today. So, Narika, let me try launching the poll. Okay, so Narika, if you could please launch the poll. It seems like it's not possible from my computer. So Narika,
Yes, is the poll visible? It's launched
yeah.
from my side. Yes, I see people.
Okay, great.
Yes. So my first question is, what is your level of education, or in that matter, what is your designation? Are you a high school student, an undergrad, or a grad? Undergraduation, that means bachelor's, graduation, master's, post-graduation, or a PhD fellow or a postdoc fellow? And also, I would want to know about your research interests. So what are your research interests? Machine learning, data science, infectious diseases, or cheminformatics, genomics, transcriptomics, metagenomics, epigenetics, or the general bioinformatics? And also, we would want to know if you have signed up on the Omicslogic Learn portal. And if yes, it's great. If not, then I would be sharing the link with you, and you can go ahead with signing up and creating your profile. Thank you everyone for answering. I would keep the poll for more 30 seconds. Thank you.
Okay, thank you, Sonalika. And while you're completing that, there's just a couple more things that I wanted to share. Okay, so what I wanted to add on to this is that in the process of getting started with data science, but also getting to a level of understanding, as we already heard from Clinton, one of the highest levels of understanding is when you can meaningfully apply the methods and skills that you have acquired to a project where you can demonstrate not just that you've memorized some scripts, but that you actually understand how to extract meaningful signal, interpret it in biological terms, and be able to present it answering questions to experts and others, peers like you. So to do that, the Omicslogic portal incorporates several features that will help you do just that. The first one is that you will gain access to these technical skills that you can practice on given examples. But what if you want to try an example of a different kind? And so the first thing that I want to show you is that beyond just the technical coursework, you will also have access to project examples. And these include examples in cancer. They include different types of data from cell lines, from animal models, as well as from patients. That also includes some clinical information. You can also look for infectious diseases, where you will find information on COVID-19, Ebola virus, and others. You will be able to find many other types of examples that include agriculture, biotechnology, neuroscience, and many other examples. But beyond that, all of the methods and all of your activity on the site is geared towards allowing you